Hey guys, welcome back to SciXShy. In this lesson, we're going to be talking about protein synthesis. And this is the beginning of our chapter on gene expression. So when we talk about protein synthesis, we have two different processes that kind of combine to create your protein. You have transcription, which is changing DNA into RNA and moving it out of the nucleus. And you have translation, which is translating your RNA into the new type of macromolecule, your protein. And so your protein is made up of your amino acids. And if you want to review amino acids and proteins or nucleic acids just to get a refresher, make sure to check out our series on macromolecules. So now that we've kind of gone over a summary of transcription and translation, let's get into the meat of transcription. Now, when you look at what I've drawn here, there's a lot of words. So let's just focus on your DNA. You have your DNA strand, and what occurs is you have your RNA polymerase, which opens up the DNA, allowing your template strand and your coding strand to be exposed. And so these two are separated. They used to be hydrogen bonded together. Again, that's in our nucleic acid video, but they're hydrogen bonded together. And these are broken by the RNA polymerase. And polymerase basically meaning it produces the polymer, which is our mRNA. And now RNA polymerase also plays the important step of attaching the next base pair to your RNA strand. And this begins with your promoter. So your promoter is a chain of nucleic acids that kind of start the production or start the creation of your mRNA strand. And so you begin with your promoter, and as you go down each of the base pairs in your template strand, you have another mRNA or RNA base pair that matches that. Now, we've gone over this in past lessons, so I want to give you the chance of guessing what our mRNA strand is in this case. So again, your template strand is AGCT, GCAG. And if you want to write this down or just think about it, what is our mRNA strand? And I'll give you a second. So hopefully you've paused the video, but our mRNA strand is uracil. And again, uracil and adenosine are pairs when you come to mRNA or RNA in general, while in DNA, adenosine and thymine are pairs. And so you have, again, adenosine matches with uracil. So this is our RNA strand. You have uracil, cytosine, guanine, adenosine, cytosine, guanine, uracil, cytosine. So now that we have our mRNA strand, which is linked together by the RNA polymerase, let's talk about your template and coding strands. And what are these? So as these two strands are broken apart by your RNA polymerase, you can see that the mRNA is actually closer or more similar to your coding strand. The base pairs that are in your mRNA strand are more similar to your coding strand. And this is because your template strand is only there as a template, as pair of the coding strand. And so it allows the RNA polymerase to code for the coding strand in the mRNA. So the code for these proteins are actually contained in the coding strand. And so I would just want to make that clear. Another key factor is the difference between monocistronic and polycistronic. And this is key to when you understand the difference between eukaryotes and prokaryotes. So eukaryotes are actually monocistronic. And what this means is that each mRNA codes for only one polypeptide, while polycistronic prokaryotes, and so each of the polypeptides are actually coded within a single RNA molecule. So this is specific to prokaryotes, and again, eukaryotes are monocistronic, and that's what we're doing here. You have a single polypeptide that's coded for in a single mRNA strand. Now, your RNA polymerase stops coding for this mRNA once it hits a stop codon. So in the mRNA side, that would be uracil, adenosine, UAG, or UGA. And these are your stop codons. And so once it stops coding for this, your DNA is brought back together, and your RNA has one more step before it leaves the nucleus. Now, what is this step? It's called RNA splicing. In your DNA, you have coding and non-coding fragments. And so these are called introns and exons. So your exons are coding fragments. So they code for the actual protein, while introns are just there as non-coding fragments. They have other responsibilities or other properties, but we'll be talking about that later or in higher levels of biology. But just in this case, you have introns and exons, and your exons are coding fragments. And so your RNA transcripts all of this. It transcripts both your introns and your exons. And so when it gets to the splicing stage, which is specific to eukaryotes, what happens is your exons are chosen. So specific exons are brought together by your spliceosome, which is a protein that is key when you are talking about RNA splicing. So your spliceosome puts together these different exon splices and creates your final mRNA strand. And this mRNA strand is also capped by your GTP cap and your poly A tail. And so your poly A tail is on your 3' end and your GTP cap is on your 5' end. But this is your final mRNA, and it only ends once you get to the splicing stage. And so your same mRNA can be cut and restitched into different combinations, and so this kind of helps in variation in that single mRNA strand. So we have our final mRNA strand, and now it can finally leave the nucleus. All of this happens in the nucleus. And so this wraps up transcription.